Hello everyone, this is Dr. Leili Hatami. Welcome to the Human Anatomy course, Chapter 2, Cells, the Living Units, Part 2. In Chapter 1 video, we discussed about the plasma membrane, its structure, and general overview of the plasma membrane function. And now we are going to continue the cell's structure. So as we already know, this cell has three main components: the plasma membrane, the nucleus, and then the cytoplasm. Now we are going to study the cytoplasm. Cytoplasm is everything that lies between the cell's outer boundary, which is the plasma membrane, and the nucleus. Within the cytoplasm, you find different components that work together to keep the cell functioning. The cytoplasm consists of cytosol, organelles, and inclusions. Let's start with the cytosol. Cytosol, also known as cytoplasmic matrix. The cytosol is a jelly-like fluid within the cytoplasm. It's a fluid where all the other cellular elements are suspended. Cytosol is mainly made up of water, ions like sodium, potassium, calcium, and enzymes which we need for facilitate the chemical reactions taking place inside of the cytoplasm. Then next we are going to study the organelles. Organelles are the little organs which all the thing that the cell is doing is because of the organelles function. The first organelle we are going to discuss is ribosomes. Ribosomes are like tiny factories inside your cell that they're going to make protein. So they are the site of protein synthesis for cellular and extracellular function. As you can see in this picture, each ribosome is composed of two subunits, small subunit and large subunit, which each of them is composed of proteins and a type of RNA called ribosomal RNA. There are two types of ribosome found inside of the cell. We have free ribosomes and bound or attached ribosomes. Free ribosomes float around in the cell's fluid, making proteins that will stay inside the cell, and the bound or attached ribosome, on the other hand, are attached to a structure called the endoplasmic reticulum, and they are going to make the proteins that are either sent outside the cell or inserted into cell membrane. So doesn't matter if they are free ribosome or attached ribosome, they are going to make the protein. The whole process of making proteins from the instruction in RNA is called translation. So ribosomes are basically the places where the cell follows the recipes in RNA to build the proteins it needs. So literally means that the process of conversion of mRNA or messenger RNA to the protein is called translation. The ribosome is involved in translation. Next organelle is the mitochondria. Mitochondria's main job is to produce the energy for cellular function, specifically a molecule called ATP. So the mitochondria is the ATP or energy production factory or machine. Mitochondria is the most complex organelle in a cell, and they have the only organelle, they have their own DNA. If you look at the mitochondria, or if you want to draw the mitochondria, you have to draw two membrane for mitochondria, the outer membrane and the inner membrane. And then inside we have the matrix, where the process of making ATP happening here. And also in the matrix of the mitochondria is the place that you can find the mitochondria's own DNA, which, as I mentioned before, it is the only organelles inside of the cell which they have their own DNA or genetic material. Then we discuss about where the ATP is going to be, the ATP generation starts, which is going to be the matrix. And then because the mitochondria is responsible for producing the energy, the cells that need a lot of energy, like muscle cell, for cellular contraction and relaxation, they need energy or ATP. The cell like sperm for movement needs energy, and also the other cells, also all the cells needs energy, but these cells actually needs a lot of energy. Or the cells that they are doing lots of the active processes for membrane transportation. For example, the cells can be found in the kidney tubules, and they are active in the membrane transportation across the plasma membrane through the active transportation. They need a lot of energy, so they must have bunch of these mitochondria, because the mitochondria is the energy factory. Next organelle is called endoplasmic reticulum. There are two types of endoplasmic reticulum inside of the cell. We have rough endoplasmic reticulum, or RER, and then the next one is smooth endoplasmic reticulum, or SER. The rough endoplasmic reticulum is like a factory with tiny workers, as the ribosomes are attached to its outer surface. And now, guess what's gonna be the rough endoplasmic reticulum main function? Because ribosome is a protein factory or protein making machine, so rough endoplasmic reticulum is primarily involved in the synthesis of protein. Then the cells that are producing the enzyme, which they are protein, or the hormone as a protein hormones, they have lots of ribosomes, and also they have lots of rough endoplasmic reticulum. Let me make a real example. In the organ pancreas, which is one of the organ of digestive system, the pancreatic cells, they are producing the digestive enzymes. And enzymes are protein. Okay, and guess what? In the cytoplasm of the pancreatic cell, we should have lots of rough endoplasmic reticulum and also ribosomes. Okay, let's continue with the second type of endoplasmic reticulum, which is the smooth endoplasmic reticulum. Picture a network of tubules without those ribosomes. This is the smooth endoplasmic reticulum. It's more into lipids, making lipid, metabolizing lipid. So it means that the smooth endoplasmic reticulum are not involved in protein synthesis. They are involved in lipid synthesis and also metabolism of the lipid. We also can add the smooth endoplasmic reticulum helps detoxify the harmful drugs, and also in some cells they are responsible to store the calcium ion. Again, let's make an example. Um, let's make an example of the hormone, which they are steroid, kind of the lipid, like the sex hormones, testosterone hormone. Testosterone hormone is the male sex hormone, which is a steroid, kind of lipid. And there are some cells inside of testes, we call them as Leydig cells, which are responsible to produce testosterone. Okay, so as the testosterone can be stored or lipid, so it means that inside of the cells, the Leydig cells, we must have lots of smooth endoplasmic reticulum. Or another example, the fat cells or adipocytes, they also have lots of smooth endoplasmic reticulum because of they have the lipid synthesis and also the lipid metabolism. And guess what? Which organ do you know is responsible for detoxification? Good job, liver. So in liver, there are some cells, we call them as hepatocyte, as hepato means liver and cyte means cell. The hepatocyte, they have lots of smooth endoplasmic reticulum as they are responsible to detoxify the lipid-soluble drugs. And what about the cells? Their smooth endoplasmic reticulum is going to store the calcium ion. Just remember that the muscle cells, they need the calcium ion for contraction. Okay, and then inside of the cytoplasm of muscle cell, there are lots of smooth endoplasmic reticulum, which they are specialized or modified to store the calcium ion. So as you can see in this picture, you can find this smooth endoplasmic reticulum here without the ribosome attached. So as I mentioned, is into the lipid synthesis, lipid metabolism, and in some other cells, they are doing detoxification of the lipid-soluble drugs and also storing the calcium ions. And in this side, you can see these rough endoplasmic reticulum, which these little red dots are ribosome, and they are responsible for protein production because of the ribosome. The next organelle is the Golgi. Golgi is like the cell's packaging and distribution center, ensuring that products from the rough endoplasmic reticulum get processed correctly and sent to the right places within and outside of the cell. So it means that there is a close relation between the rough endoplasmic reticulum and Golgi apparatus. It means that the protein which is going to be made in rough endoplasmic reticulum, it is not a final product. Then this product of rough endoplasmic reticulum should be sent to the Golgi, which inside of the Golgi, they're going to be modified and also packaged, and then they're going to have 3 different destinations. Or simply we can say the Golgi is like the cell's post office. When the Golgi apparatus gets product from the rough endoplasmic reticulum, it processes, packages them. Then these packages sent to their proper destination in the cell. The first destination, you can see that it's going to be the secretory vesicle, which they're going to be released by exocytosis, mostly in gland cells. The second destination is the vesicle membrane fuses to the plasma membrane, and it's going to be part of the plasma structure. And the third pathway or destination would be it's going to make a sac with digestive enzyme, which is called lysosome, and it can happen in all cells. And let's review the Golgi function in this picture. So as you can see, the Golgi complex is going to be found very close to the rough endoplasmic reticulum, and then the product of rough endoplasmic reticulum is going to be sent to the Golgi. And then after processing and packaging the protein and all the product, then it's going to be sent to the pathway 1, which mostly is happening in gland cells. So as you can see in the gland cell, these vesicles, which vesicle is a sac, gonna be like surrounded by a membrane, and now inside of this sac or the vesicle, it's gonna be like the product of rough endoplasmic reticulum and Golgi. Can be a hormone, can be an enzyme, then or anything that a gland cell needs to send them out. So then vesicles are released by exocytosis, which is a process where the vesicle membrane fuses with the plasma membrane and the contents are released outside of the cell. Okay, and then the second pathway, as you can see, the product of rough endoplasmic and Golgi complex going to be sent to the plasma membrane, and then it's going to be fused with the plasma membrane, going to be part of the plasma membrane. That is the second pathway. And the third one, picture another type of vesicle called a lysosome, as a sac filled with digestive enzymes. Lysosomes are like the cell's garbage disposal units. They break down waste material and old cell parts, keeping things tidy inside of the cell. Lysosome is another organelle found in the cytoplasm or the cell, but it's going to be made by Golgi, or it's a product of RER and Golgi. So RER is producing those digestive enzymes, and Golgi is going to modify those enzymes and package them as this sac of digestive enzyme, which is called the lysosome. So do not forget, the lysosome is an organelle, but the origin of lysosome is the Golgi complex. Okay, so this is the three pathway of Golgi in packaging the product of rough endoplasmic reticulum. Then let's continue with the other organelles, which they are membrane bound and gonna be found in the cell, and they have very important function as well. Imagine lysosome as small sacs filled with a special set of digestive enzyme inside. They digest unwanted stuff like old or damaged part of the cell, worn out proteins, and other materials that need recycling. So just draw a sac, membrane bound sac, which inside of this sac or vesicle or this organelle, we have bunches of the digestive enzymes, which this enzyme or hydrolytic enzyme can digest or break down the unwanted substances, damaged or worn out proteins or organelles found inside of the cell. And yes, which cells, they need to have bunches of the lysosome. You are right. Phagocytic cells. We discussed about the phagocytosis and phagocytic cells a little bit in chapter 2 part 1 video. Phagocytic cells like white blood cells, the macrophages, they need lots of lysosome to degrade the ingested bacteria and also viruses. Then next organelle is the peroxisome. Peroxisome is very similar to the lysosome. If I want to draw the peroxisome, I should draw exactly or the similar structure like the lysosome. A membrane bound sac with set of enzyme inside. But what is the difference between the lysosome and peroxisome? Their enzymes are totally different. The enzymes inside of peroxisome called oxidase and catalase, which they are responsible to neutralize the free radicals and break down the harmful substances or poisons or toxins. So it means that they are kind of very similar to each other, but their components, their enzyme components, and also their function are different. So again, in which cell we can find like lots of peroxisome in the liver and kidney cells, because liver is going to detoxify the toxin, poison. So then we can find lots of the peroxisome in hepatocyte or liver cells and also in kidney cells. And then we can also add the peroxisome are also responsible for breaking down the long chain of fatty acid in lipid metabolism. In general, we can say the lysosome are the cell's cleanup crew, while the peroxisome are like detox center for the cell. So to make sure that the cells stay healthy and free from the unwanted substances or even toxin or poisons. Next organelle, we have the cytoskeleton. The cytoskeleton is the cell's own skeleton. Is a network of rods providing support and also allowing the movement inside of the cell or for the cell. The cell skeleton or cytoskeleton has three types of rods. We have microfilaments, which are the tiny filaments made of a contractile protein called actin. Just like muscles in your body can contract, these microfilaments can change their shape, helping the cell to move and maintain its structure. The next is intermediate filament. They are a thicker protein fibers in comparison with the microfilaments. Their main job is to provide the structural stability for the cell, and they act like the cell's sturdy cables holding everything in place. And the last one, which is the thickest one, is the microtubule, which these are the cylindrical structures made of proteins. Microtubules act as the cell's highways, providing tracks for moving things around inside the cell. They also play a role in separating the chromosomes during the cell division. So in simple terms, the cytoskeleton is like the cell's supportive framework made of microfilament, intermediate filament, and microtubules, allowing the cell to maintain its shape and perform various movements, and it's the cell's internal scaffolding. In this picture, you can see these three types of cytoskeleton. The thinnest one, which has 7 nanometer in the thickness, is the microfilament, and then in the thickest one, which has 25 nanometer thickness, is called microtubules, and in between we have the intermediate filament. Next, we have the centrosome and centrio. Centrosome is a spherical structure in the cytoplasm composed of the centrosome matrix and centrioles. So as you can see in this picture, this is spherical structure which we zoomed in, and then you can see here, this is the centrioles, these two structure here, and then you can find the centrosome matrix around it. The total is called the centrosome. But what are these structures, as we call them as centrioles? Centrioles are cylindrical structure, which each of them is composed of 27 microtubules. We just talked about the microtubule is one of our cytoskeleton, and then now you can see how the microtubule can be involved in making the centriole. So one more time, the centriole are the cylindrical structure, each made up of 27 short microtubules, and they are like the cell's construction workers. Centrioles are involved in a few important tasks. They play a role in forming the cilia. What is the cilia? Cilia is the hair-like structure found on the cell surface, which they they can move, and with their movement they actually can move the secretion or like the mucus gonna be found at the surface of the cell. And also they're gonna be involved in forming flagella, which is like a like a sperm tail, which they are using for movement of the whole cell, like a sperm cell. The only cell in the human body that they have flagella is the sperm cell in the male body. Okay, and also the centriole gonna be involved in forming the mitotic spindle, which we're gonna study the mitotic spindle in the next part of the chapter 2, which is critical for cell division. So please make sure you know that centrioles are the cytoskeleton, which they're gonna be important in forming the cilia, flagella, and mitotic spindle. And last part of the cytoplasm, or the component of the cytoplasm, is inclusion. So far we talked about the cytosol and organelles, and now we are going to talk about the inclusions. Cytoplasmic inclusions are temporary storage in the cells depending on the cell's need, and they are not present in all cell types. The pigments, crystals of protein, food stores, they are the inclusion. They do not have a metabolic function. So as I mentioned before, is a temporary storage or temporary structure. Let's make an example of pigments. Imagine a skin cell in your body. Within the cytoplasm of this cell, you might find melanin. Melanin is a pigment which is actually the inclusion. The melanin pigment is responsible for the color of your skin, hair, and eyes. Or as a food storage, we have lipid droplets, which gonna be found in the liver cell and fat cells. They're serving as a temporary storage for energy. Glycosome is another form of storing the sugar. These structures are present also in the liver cell, as glycogen is stored as a source of energy. So the glycosomes is the storage unit for glycogen, and they consider as inclusion, as they do not have a metabolic function. They can be temporary. Sometimes we can see them, sometimes there are no, for example, glycosome or lipid droplets in that cells, or not all of the cells in our body they have pigment melanin, so they can be found in some cells but not in all type of cells. Okay, so that was the ending part of the part 2 of our chapter 2. I'm going to continue the next part, which we're going to talk about the nucleus and cell division in part 3 of chapter 2. Thank you for watching.